Each one of our cells contains a complex transcription and translation process that turns DNA codes into proteins. However, what happens when this process makes a mistake? Let's take a look at how the insertion of one extra base pair results in the production of a truncated non-functional protein. So in this example, we're going to use these nice cortical game pieces to represent our different bases. The green one will be G, purple will be C, orange will represent U, and the red will represent A. Here we have an mRNA sequence where every 3 base pairs codes for the shown amino acids. This is the beginning of the sequence for the normal protein. What happens when an extra base pair decides to insert itself into the sequence? Let's say an adenine nucleotide inserts itself into the mRNA sequence. What does the cell do now? If the mistake is not caught and fixed, the normal transcription and translation process will continue. However, a much different protein is going to be made. The first 2 amino acids are normal, but instead of coding for phenylalanine at the 3rd codon, the mRNA codes for isoleucine, and so on. In this example, the protein is cut short because the 5th codon is a stop codon, therefore resulting in a mutant protein. Once a stop codon is present, the remaining base pair sequences no longer matter. So how do you figure out the mRNA sequence when you are given the amino acid sequence of the normal and mutant protein? One way is to make a table like this one showing all the different codon options and then looking for a difference. The first difference is right here. Here we can see that an A was added in, so this must be an insertion. You can then figure out the rest of the sequence by looking at what is given. The original code has at least 2 U's, so there must be 2 U's after the inserted A. The next codon has to start with a C, as shown here, so UUC must be the mRNA sequence for phenylalanine. You then continue with the same back and forth till you have used all the information given. At this point, there is not enough information to determine the codon for lysine or glycine yet. Once you have done this, you are able to then deduce what the mRNA sequence is.